Hi everyone, welcome back to my channel. Today we are going to explore a fascinating scientific technique called chromatography. Whether you are a student or a scientist, or just curious about how scientists separate mixtures, this video is for you. Let's get started. So here is our first slide. What is chromatography? In simple terms, chromatography is a method used to separate different components in a mixture. Imagine you have a mixture of different colored dyes. With chromatography, you can separate these colors to see what individual dyes make up the mixture. This technique is super useful in many fields like pharmaceuticals, environmental testing, the food industry, and even forensic. For example, forensic scientists use chromatography to identify substances found at crime scenes. Here is the another slide. How chromatography works. Now let's talk about chromatography works. There are two main parts of the process, the mobile phase and the stationary phase. The mixture dissolved in the fluid is called mobile phase. The mobile phase carries the mixture through structure holding another material called stationary phase. Different components of the mixture moves at different speeds, causing them to separate. Now let's talk about the mobile phase and stationary phase in details. Mobile phase. Think of this as the fluid that moves. It can be liquid or a gas. Imagine you have a stream of water. This is our mobile phase carrying leaves which represent the components of our mixture. And the other one is stationary phase. This is a material that stays still. It can be a solid or a liquid spotted on a solid. Using our stream example with rocks and sand where the leaves might get caught. When we put our mixture into the mobile phase, the components travel through the stationary phase at different speeds. This is beneficial. This is because each components interact differently with the stationary phase. Some parts might stick more and move slowly, while others zip through the quickly. This difference in movement helps separate the components. Here is the another slide for the steps in chromatography. Let's break down the process into simple steps. The first step is prepare the sample mixture. Imagine we have a mix of different colored inks. Introduce the sample into the mobile phase. We dissolve our ink into a solvent like water or alcohol. You can use any which acts as our mobile phase. Third step is mobile phase carries the sample. We place the strip of paper or stationery phase onto the solvent. And the fourth step is component separation. As the solvent travels up the paper, it carries the ink with it. Different ink moves at different speed and starts to separate, detect and analyze. This is our last step. After a while, we see different colors spread out on the paper. Each color represents a different components of our original mixture. Here are the types of chromatography. There are four types of chromatography. The first one is liquid chromatography, second one is gas chromatography, third one is thin layer chromatography, fourth one is paper chromatography, liquid chromatography. It uses a liquid mobile phase. For example, in lab, scientists might use LC to separate vitamins in a supplement to check their purity. Gas chromatography uses a gas mobile phase. This is often used to analyze gases in the atmosphere or to test the purity of substances that can be vaporized without decomposing the essential oil. Thin layer chromatography uses a thin layer of material like silica gel on a glass plate as the stationary phase. It is quick method used in labs to see if reaction has occurred by comparing the movement of substances on the plate. Fourth and last one is paper chromatography. Uses the paper as the stationary phase. This is a simple and common method used in classrooms to teach basic concepts of chromatography. For instance, you can separate the pigments in a leaf to see the different colors it contains. Now let's move toward the application of chromatography. Chromatography is incredibly useful. In pharmaceuticals, it is used to ensure drugs are pure and contain the correct ingredients. Imagine you are developing a new medicine, you need to make sure it has exactly what it should and nothing else. For environmental testing, chromatography helps detect pollutants in water, air, and soil. Scientists can find out is if there is any harmful substances in our environment. In the food industry, it is used for quality control. For example, ensuring that the vanilla flavor in ice cream is pure and doesn't contain any harmful additives. In forensic, this is one of the coolest applications. Chromatography helps to identify substances at the crime scene, such as drugs or toxins, which can be crucial in solving cases. Summary: to sum it up, chromatography is a powerful technique for separating mixtures into their individual components. It involves a mobile phase that moves the mixture and stationary phase that helps separate the components with various types like liquid chromatography, gas chromatography, layer chromatography, and paper chromatography. This technique has numerous applications in different fields. Thanks for watching. I hope you find this explanation of chromatography helpful, interactive, and interesting. If you have any question, feel free to leave them in the comment section below. Don't forget to like, share, and subscribe for more videos on scientific techniques, experiments. See you next time. Bye bye.